All right. Let's talk about membrane structure in this series. The phospholipid bilayer, the cell membrane, the plasma membrane, all three of those words mean the same exact thing. We're all talking about the same structure, which is this double layer of phospholipids. And we'll look at what a picture of a phospholipid looks like in a second, but this is a membrane that goes around every cell inside our body and every cell out there, period. Because if you remember when we were talking about eukaryotes, prokaryotes, both eukaryotes and prokaryotes all had a cell membrane. And cell membrane plays a very large role because it controls what goes into the cell and what goes out of the cell. And it maintains this barrier that makes it so that not anything can just get in when it wants, not anything can just get out, which is very good because our cells like efficiency. As always, you've heard me say that many times over the course of this semester already, that efficiency is key. Well, when we look at the phospholipid bilayer, there's a few things that we're going to see in them, and we'll go through each one of these things individually throughout the course of this slideshow. When we talk about the cell membrane, we say the model that scientists or biologists tend to use when describing it is what's called a fluid mosaic model. Now, let's think of all of those words together. Fluid means that something's constantly changing. It's something that it's not solid, it's not stable, it's something that can adapt and change depending on the situation. A mosaic, if you look at a stained glass window, usually you consider those mosaics because you have tiny pieces of colored glass that when you put them in together, they make up one picture. Well, your cell membrane is kind of like that because we have a bunch of different components in that cell membrane that make up the entire cell membrane. And a model is a representation. So when we talk about the cell membrane in biology terms, they kind of always refer to it as a fluid mosaic model. Now, this term right here, selective permeability, is an extremely important idea when we're talking about the cell membrane. That means that the cell controls what goes in and what goes out, or it decides what's going to come in and what's going to come out. It's selectively permeable. If I were standing out next to a fence and I throw a marble at the fence, there's a good chance a marble might get through one of the holes in the fence. If I throw a watermelon at the fence, however, that watermelon's probably going to hit the fence and fall backwards, or possibly break, and that would be kind of fun to look at, but the watermelon wouldn't be allowed to go through because it doesn't fit. Well, that's sort of how our cell membranes work around with this. Phospholipids and proteins can all move around in the layer. They're not stuck in one spot. They can change places with other things within the cell membrane, and there's specific passageways that allow things through if they can't get through the cell membrane. So even though the watermelon can't get through, there might be a gate somewhere in that fence where you could walk the watermelon through. Well, cell membrane kind of works the same way. There are gates in the cell membrane that we'll talk about over the next couple lectures. When we look at a phospholipid, we have these blue circles up top right here. Those are our phosphate heads. Phosphate heads have a polar region, which is going to make them hydrophilic, meaning they're going to interact with water. In the middle of the membrane, we have these lipid tails. So right here, I'm going to go around one phospholipid. I'm hoping you can see my highlighter as I'm doing that. But that one guy right there, the one blue circle and the two gray legs, is going to be one single phospholipid. So I got one layer of phospholipids on top, one layer of phospholipids on bottom. That's where this whole phospholipid bilayer comes in, because bi means two. And when we talk about lipid tails, well, hopefully you remember from macromolecules that lipids are going to be hydrophobic. Now, that's hugely important that we have this massive hydrophobic layer surrounding every cell in our body. That layer is going to make it so that water and anything that dissolves in water can't just cruise right through the phospholipid bilayer. It can't just go right into our cell membrane. And I mean, think about it. We have ions dissolved in water, we have nutrients, we have other bigger molecules, proteins, all kinds of things dissolved in water that have to figure out a way to get into the cell. And that membrane controls how things get in and out of the cell. When we talk about proteins, the proteins are embedded all throughout the membrane, and proteins, we'll go a little bit more into them a little bit later, but the domain is what part of the membrane is holding onto that protein, or what part of that protein is holding onto the membrane. Now, if you look at this picture on the right hand side over here, this protein is going all the way through the cell membrane. Now, part of that protein's touching these phosphate heads. So hopefully you remember that side chains on the protein determine how it folds. And hydrophilic or polar side chains are probably going to be the ones on the edge of the membrane. In the middle of the membrane, where we have a bunch of lipid tails, this is probably where we're going to find all of our hydrophobic R groups right there, so that they can interact with the lipid tails. And again, more hydrophilic and charged or polar R groups down here on this bottom end of the cell membrane in that protein. So that structure of the membrane kind of determines what can sit in there and where it goes. Now, carbohydrates. When we look at this picture on the right, the carbohydrates are these green chains that are attached to the cell membrane. Carbohydrates are always going to stick outside the cell, and they're going to do one or two things, and we'll sort of go over those two things in a little bit. But you're going to find these things, again, carbohydrates are chains of monosaccharides that are attached to these things. This one's attached to a protein, so it's something that we're going to call glycoprotein in a little bit. We also find cholesterol in the cell membrane. It's these little yellow guys that look like kind of cobs of corn. And look where the cholesterol is hanging out. Cholesterol is hanging out right in the middle of all the lipids. Well, that's because cholesterol is a lipid. It's made of long hydrocarbon chains, so it's going to find itself at home in those lipids, and that's going to help with stability of the membrane. When we talk about the cell membrane and how thick it is, it's really not very thick at all. You're talking 8 nanometers. That's way thinner than a hair. So, I mean, these things are very, very tiny and very important for the cell and its function. A couple things that really affect how the membrane works is, one, how many lipids there are. More lipids is going to make it a little bit stiffer. More cholesterol is going to make it a little bit more stiff. And then temperature, well, colder temperatures make the membrane a little bit more stiff also. And think about if you've ever gone outside and tried to go running when it's cold out. You kind of got to take a little bit more time to warm your body up and get your muscles moving to generate some heat so that they can start moving efficiently. Whereas if you do that in the summertime, you pretty much walk outside and you're ready to roll. Well, what's your body made of? It's made of a bunch of cells. Your muscles are all cells and they need to get going. So you need to get the temperature up to the right spot so that the membrane is a little bit more fluid and a little bit more apt to maneuver you through space. Lipid composition is going to vary drastically. Cholesterol amounts can vary, and when we talk about lipid composition, lipids come in two forms if you remember from the macromolecule section. Saturated, which are going to stack on top of each other really nicely, and unsaturated lipids, which have at least one double bond somewhere in their chain. That makes them a little bit more fluid, a little bit looser. Lipid composition also is going to help determine the rigidity of that membrane. More lipids is going to make it more rigid, especially more cholesterol is going to make those membranes a little bit more rigid. Sorry, having trouble talking today. Again, just sort of rehashing the temperature thing. In cooler temps, the fluidity of that membrane is going to be a little bit less. It's going to be a little bit stiffer. When animals are about to hibernate, they start to store a lot more cholesterol and a lot more lipids in their membranes, and their temperature is going to drop to help maintain their membranes throughout the time that they are hibernating and sleeping for weeks at a time or months at a time. Now, we're talking about proteins found in the membrane. There's 3 types of proteins found in the membrane. First one we'll talk about is peripheral membrane protein. Now, a peripheral membrane protein, if you hear that word peripheral, think of your peripheral vision. Your peripheral vision is what you see off to the side when you're looking straight ahead. Well, a peripheral membrane protein is one that's not attached to the membrane. It's attached to most likely another protein that's in the membrane, and this peripheral one is hanging off to the side, sort of like your peripheral vision is off to the side. So this one's not really embedded, but it is there, and it might be helpful for signaling or moving a message within the cell somewhere else. And when we talk about G proteins, we'll talk about some peripheral membrane proteins. Integral membrane proteins are integrated into the cell. They're put in the membrane somewhere. Sometimes they go all the way through, sometimes they don't go all the way through. It just depends on the membrane protein. And again, we look at the R groups to figure out where that protein, where the different amino acids might interact with the cell membrane. The last kind of protein is what we call a transmembrane protein. And a transmembrane protein is one that has an opening on the outside and the inside of the cell. And these ones are going to act as tunnels, usually, so that things can get in and out of the cell. Again, the phospholipid bilayer doesn't allow everything in, so the transmembrane proteins kind of act like a side door that you can go in to get past that cell membrane. Last side we'll look at today is the different carbohydrate types that we find. Carbohydrates can come in one of two forms, glycolipids or glycoproteins. If you see the word glycolipid, hopefully you know glyco means sugar and lipid refers to lipid. So this is a carbohydrate that's attached to a lipid in the membrane. And carbohydrates are always going to extend outside the cell surface, so into the extracellular space. The glycolipids are going to be big ones to be help cells attach to each other so that they can hold on to each other and not get separated. It has to do with cells having a little bit of give in that extracellular space that we talked about in the last lecture. Glycoproteins is a sugar attached to a protein. So if you have a transmembrane protein with a sugar sticking out of it, that carbohydrate that's sticking off the side is going to act as a receptor. So that molecule might be in the extracellular space attached to that carbohydrate and tell the protein that's the transmembrane protein, hey, open up so something can get inside of you. And that sort of has to do with cell communication, which is something that we'll start going into very soon here. So that does it for what's involved in the structure of the membrane. Next one we should be talking about is cell communication. Have a good afternoon.